Okay, a pleasant morning to all. Actually, it's a rainy morning. I am Panpilo Di Guzman, and I will serve as moderator for today's webinar session. Welcome to the webinar series on Biotech Innovations for Sustainable Agriculture. This activity is a build-up activity for the NBW, organized by DA Biotechnology Program Office, ISAAA Inc, DA Bureau of Animal Industry, Biotech Coalition of the Philippines, and Corteva Agriscience. We currently have around more than 400 participants representing different stakeholders, and others are still joining in. Today's webinar is part of the 3-day series that explores the role of biotech innovations in driving agricultural sustainability, providing solutions to some of the most pressing problems in the agricultural sector and challenges surrounding it. Before we start, a few house rules. This webinar will run for 2 hours and will be recorded for documentation purposes. The webinar will also be streamed live on ISAAA's Facebook page and YouTube account of ISAAA, and I think it will be also run in DA Biotech program's website. Make sure that you have good internet connection so you will not miss any of the discussions. Please turn off your camera and your audio muted during the presentations to avoid any distractions. We encourage interaction exchange, thus we welcome questions from the participants. Please use the Q&A button at the bottom of your screen for any questions and clarifications you may have. Please indicate to whom you are directing the question. We will entertain the questions during the open discussions. And we will ask you to answer a post-webinar survey on day 3. So make sure that you complete the 3-day webinar series, and this will be the basis of the issuance of your certificate of participation. Thank you very much for taking your time in attending this webinar, and we look forward to your active participation. Now for the opening remarks, Dr. Aldimita will give us the opening remarks. She holds a PhD in botany from Purdue University, USA, and postdoctoral fellowship on golden rice and Albert Ludwig University in Germany. She has served as Chief Science Research Specialist at the Biotech and the Biotechnology Coordinator at Phil Rice and former researcher at Erie. She leads the development and publication of the annual Global Status of Personalized Biotech Crops or ISAAA, which hopefully will be continued next year. Dr. Aldimita has published 31 papers in scientific journals and proceedings and chapter in 2 books on biotechnology. She is the recipient of various awards, including the Outstanding Young Scientist by NAST, the NAST and Italy-based the World Academy of Science Prize in Biology, 10 Outstanding Women in Nation Service or from Towns Foundation, Most Outstanding Researcher, Senior Researcher at Phil Rice, and Filipino Faces of Biotechnology. And of course, the 2020 UPLB College Distinguished Award for Excellence in International Service and Cooperation. You have now the virtual mic, Dr. Aldimita, for your opening remarks.
Thank you so much, dear Popen, and welcome everyone to the webinar series on Biotech Innovations for a Sustainable Agriculture, a 3-day build-up activity for the 19th National Biotech Week with the theme Empowering Innovation for Sustainable Future with Biotechnology. The webinar is hosted by us, the International Service for the Acquisition of Agri-Biotech Applications, Incorporated, a nonprofit international organization working to feed the world with knowledge through the packaging of biotech information, development of science communication modalities, facilitating knowledge sharing, capacity building, and assisting domestic biotech programs. At this point, I would like to recognize the organizing team composed of Charmaine Lopez of DA Biotech, Dr. Nina Halos and Dr. Abe Manalo of Biotech Coalition of the Philippines, Karen Panaso of Bureau of Animal Industry, and then us as hosts, and with the support of Corteva Agriscience, represented by Dr. Antonio Alfonso. This webinar series is quite timely, as efforts toward sustainable agriculture should be more than doubled in order to feed the world population of more than 10 billion by 2050. Business as usual attitude for food productivity will no longer work, as problems on population, climate change, and dwindling resources complicate all efforts towards food sufficiency. Our food producers should be equipped with new innovations developed through the continuous understanding of the problems, analysis, and development of ideas towards some proof of concept scientific solutions, implementation and verification of the solutions, and dissemination of the technology to move for adoption to create the all-important changes globally. So in the next 3 days, we will be tackling innovative technologies developed to address problems in food and agriculture. The topics for the 3 days are new breeding innovation in crops, the technologies application in mitigating pesticide use and regulation. So this is the title of the webinar today. And then tomorrow will be on biotechnology application and regulation in animals. And on Friday, we will have bio-innovations, potential and challenges, the concept of social license, and the path to commercialization. We have invited local and international scientists, and we'll devote at most 30 minutes for the question and answer portion every day to further clarify concerns. So let's take advantage of that Q&A. As I said, we had more than, Poppen said we have already more than 400 participants today. We had more than 1,000 registrants, indicating the interest of the general public, local and international, in biotech innovations for food and agriculture. Thus, we do hope that the 3-day webinar will somehow increase your knowledge of biotech innovations in crops, animals, and appreciate their potential to help build sustainable food systems. We invite you to complete the 3-day webinar series and respond to our post-webinar survey at the end of the 3rd day. This will serve as a reference for our improvement for future webinars, and your ticket to obtain a certificate of attendance. Have a great day, everyone, and happy learning.
Okay, thank you very much, Ma'am Ola, for giving us an overview of what to expect for today's webinar and for the next 3 days. So we will now call Dr. Claro Mingala to give us a key message. He is the director of the Philippine Agriculture and Fisheries Biotechnology Program of the Department of Agriculture. He was formerly the Center Chief of the Livestock Biotech Center at the Philippine Carabao Center, as well as the chair of the drafting committee for the regulation of animal biotechnology in the Philippines. Dr. Mingala received his Doctor of Veterinary Science and Medicine and Master of Veterinary Studies degrees at the Central Luzon State University, and finished his PhD in Veterinary Science at the Hokkaido University in Japan, specializing in infectious diseases. He pursued his postdoctoral fellowship in Cornell University, Ithaca, in New York in 2016 under the Fulbright Philippines Agriculture Scholarship Program for Advanced Research. Since then, he worked on the research and development of the diagnostic tools and vaccines for priority livestock diseases. He is the recipient of the 2010 Philippine Veterinary Medical Association Outstanding Veterinarian in Government Service, 2011 Outstanding Scientist, 2016 National Research Council of the Philippines Achievement Award, and one of the 2019 Paces of Biotechnology as the Outstanding Livestock Biotechnologist. Dr. Mingala is a career scientist with the rank of Scientist 4. Dr. Mingala, please, you can now read your message.
Thank you very much, Mr. Poppen. Good morning to everyone. Welcome to the first day of our webinar series on Biotech Innovations for a Sustainable Agriculture. This 3 days webinar is a build-up activity for the upcoming 19th National Biotechnology Week, which will be held on November 20 to 24, 2023 at SMC Site City, Cebu. Albeit virtual, we are thrilled to have you all here today for this webinar series to further explore the latest advancements in biotechnology and its impact on Philippine agriculture. We all know agriculture has always been the backbone of our society, providing us with the essential necessities of life, food, fuel, or even fiber. However, the challenges faced by the agricultural sector are growing rapidly, and it is crucial for us to explore innovative ways that can ensure sustainable and efficient agricultural practices. Day by day, we can notice that the global population is skyrocketing. Climate change is also posing a significant threat to our agricultural system, with extreme weather events becoming more frequent and unpredictable, and our resources are becoming increasingly scarce. In the face of these daunting challenges, biotechnology has emerged as a game changer. From genetically modified crops that are resistant to pests and diseases, to precision breeding techniques that enhance crop productivity, biotechnology innovations have the potential to revolutionize the way we produce food. Indeed, the recent progress in biotechnology presents unparalleled possibilities for tackling wide-range problems, including climate change, resource scarcity, and health and food security. Throughout this webinar series, we will delve into the various aspects of biotechnology in agriculture and explore the potential it holds for transforming food systems. For this day, we will lay the foundation by discussing the overall concept of sustainable agriculture and how biotechnology plays a crucial role in achieving it. In the upcoming sessions, we will explore specific areas such as new breeding innovations in crops, the technologies involved, their applications in mitigating pesticide use, and the regulations surrounding them. We will also dive into the realm of biotechnology applications and regulations in animals or livestock, understanding the potential and challenges they bring. And lastly, we will touch upon the concept of bioinnovation, its potential, and the challenges it faces. Here, we will also discuss the importance of social license and the path to commercialization for biotech innovations. As such, we have gathered technical experts from various fields who will share their knowledge and insights with us. They will provide us with valuable information on the latest advancements, trends, and challenges in the field of biotechnology. I highly encourage everyone to actively participate, ask questions, and engage with our esteemed speakers. We will hope that by the end of this series, we will have a clearer picture and appreciation of how biotechnology can contribute to the promotion of a more sustainable and efficient agricultural system. Muli isang mainit na pagbati at pasasalamat sa inyong lahat. Thank you very much. Sama-sama nating suportahan ang Biotech Innovations para sa isang masagana at matatag na agrikultura. Maraming salamat po. Mabuhay po tayong lahat.
Thank you very much, Dr. Bingala, for that key message. Indeed, biotechnology has the potential to revolutionize food production system, as you said. And now we will go on to our first topic on new technologies driving the future of plant breeding. This will be presented by no less than Dr. Ordonio. Dr. Reynante Ordonio of San Jose City, Nueva Ecija, first joined the Philippine Rice Research Institute in 2004 after more than 2 years of teaching at CLSU, where he also obtained his BS, magna cum laude, and MS biology degrees. He later obtained additional MS and doctoral degrees in agricultural science and postdoctoral fellowship from Nagoya University in Japan. He is currently a senior science research specialist at PhilRice and has been conferred the rank of Scientist 1 by the Civil Service Commission and DOST in May 2019. He currently serves as the project leader of the Healthier Rice Project at PhilRice, a collaborative project with IRRI, which aims to produce beta-carotene-enriched golden rice varieties and high iron and zinc rice to address the problem of micronutrient deficiencies in the country. Dr. Odornio's field of interest include plant genetic modification and gene editing. His earlier works have been published in international journals such as Nature Genetics, Nature Scientific Reports, PNAS of USA, and Science Magazine. He was also involved in crafting the Philippines policy on products plant breeding innovations through a DA Biotech Commission study group on new plant breeding techniques in 2018 and in the chair of the ad hoc technical committee working group on NBTs under NCBP in 2019. He is also recognized by the National Academy of Science and Technology as one of the outstanding young scientists of 2021. Dr. Ordonio, please, you can now share your slides. Yeah, it's still loading. Yeah, we're just waiting for it. Yeah, now.
Okay, so good morning to everyone, and thank you so much for having me here and for this lovely opportunity to talk about biotechnology. Today, I will be discussing the new technologies driving the future of plant breeding. This presentation, as requested, will focus on the new wave of plant breeding innovations and their potential impact in the future, and will also provide an overview of the current global regulatory frameworks for these tools. From the title, when we say new technologies for plant breeding, these are the things that will come to our mind. Number one is advanced genome transcriptome sequencing or genotyping tools. And this refers to the NGS or the next generation sequencing systems like Illumina, PacBio, Nanopore, which are really revolutionizing plant breeding right now because they drastically reduced the time and the cost it needs to sequence the whole genome compared to the standard Sanger sequencing. It's good to know that there's a lot of crops and organisms now with elucidated genomes because of these technologies. Number two is advanced analytical tools, which include bioinformatics in the exciting field of artificial intelligence. These technologies allow scientists to build useful databases and to mine and make full use of valuable information from the heaps of data being generated by sequencing technologies, and much more. And number three is advanced DNA synthesis technologies. We are not talking here of just short oligonucleotides for synthesis, but rather genes or even genome. Yes, it is now possible to build customized genomes from scratch, and this facilitates precision plant breeding and the rise of synthetic biology. And number four is plant breeding innovations, also known as new plant breeding techniques. PBIs refer to a continuously evolving suite of modern biotech tools that facilitate the development of a product or new plant varieties with desired traits in a way that is faster and more precise than conventional plant breeding techniques. PBIs mainly aim to genetically mimic natural variation to produce a conventional plant product that is not subject to strict GM regulation. But some of the PBIs can also insert a foreign gene from a cross-incompatible species, thereby producing a GMO. Here are the 8 PBIs that are available to the modern-day plant breeder. We have SDNs, ODM or oligonucleotide-directed genesis, cisgenesis and intragenesis, RNA-dependent DNA methylation or RDDM, grafting with GM material, reverse breeding, agroinfiltration, synthetic genomics, and other upcoming techniques. So let's begin with the concept of the SDNs or site-directed nucleases. By the term site-directed, this implies that this technique is targeted and specific, which is a far cry from conventional breeding technique. There are actually different SDNs like zinc finger nucleases, TALENs, and CRISPR-Cas system, and generally each differ in terms of their way of recognizing, binding to and cutting the target DNA. Now, regardless of which SDN is used, the result will be the formation of a double-strand break and recently single-strand break. And the way such break is repaired and the nature of the insert, if any, will ultimately determine how the resulting plants will be treated or classified, whether GMO or non-GMO. Now let's just have some details. Let's start with further details about oligonucleotide-directed mutagenesis. This is a form of gene editing that does not use nucleases, as in site-directed nucleases. Instead, it uses customized single-stranded oligonucleotides, which you can see on the left, designed to interfere with the normal replication of DNA in the target region or the replication fork. The oligonucleotide with the direct or desired bases, binds to the complementary region in both DNA strands in the complementary or in the replication fork rather. The cell then detects the base mismatch and starts repair, only that it is error prone, and with this, the alternative bases in the mismatch gets copied to the DNA, thereby leading a mutation. Since the mutation is just a base change, and since the non-replicating oligo introduced eventually gets degraded in the cell, there is no novel combination of genetic materials formed in the products of ODM, so they are non-GMO. And next is cisgenesis and intragenesis. These two methods involve the usual transformation vectors also being used in genetic transformation or transgenesis. The only difference with genetic transformation is that the gene insert is from a cross-compatible or same species. For cisgenesis, the gene or allele is taken from the donor unaltered, meaning it has the native promoter, coding sequence, and so on. On the other hand, for intragenesis, the gene insert is altered to some degree, meaning it may have the promoter or coding sequence of another gene or its orientation was reversed. For both techniques, since the inserted gene is just from the same species, novel combination will not be formed. The only consideration are border sequences, which are shown here in this illustration as gray bars at each end of the cisgene and intragene. And these should be considered, and these should not result in novel combination of genetic materials. And next is RNA-dependent DNA methylation. This method ultimately leads to the methylation of the promoter region of a gene, which essentially silences the gene. Methylation does not affect the DNA sequence, but rather interfering with the binding of activator proteins and transcription factors to the promoter region. So as a result, gene expression is reduced or completely blocked, a condition called transcriptional gene silencing. Now, RDDM technique may involve the integration of an inducer construct into the plant genome using genetic transformation, so resulting in a GMO. The transgene expresses double-stranded RNAs that become small interfering RNAs that will facilitate the methylation. After the transgene is bred out from the plants, the resulting null segregants will have the same genome sequence with the original variety, but have a different trait as intended. And more importantly, no novel combination of genetic materials is produced in the final product, so non-GM. And next is grafting with GM material. In this technique, a plant part is fused to an already approved GMO event. The GMO part can either be the rootstock or the scion. Now, in this technique, while the metabolites and other molecules can spread throughout the resulting chimeric plant, the DNA does not spread away from the graft junction. For instance, glyphosate resistance in the rootstock will also be observed in the scion, but the two are still genetically different since DNA is not transferred. Therefore, any product harvested from the non-GM scion, like fruits and seeds, or from the non-GM rootstock, like tubers, will not be distinguishable from and will be treated the same as those produced through conventional breeding. And then we have reverse breeding. This is a method in which we can derive parentals from a hybrid instead of the other way around. The process involves also genetic transformation of the hybrid with unknown parents to silence through RNAi some important genes involved in crossing over during meiosis. The absence of crossing over allows us to preserve the original configuration of the chromatids in gametes or haploid cells, particularly the pollen or microspores. In addition, we also have to double the chromatid through anther culture or doubled haploid technology, which essentially creates diploid plants. These will be candidate parentals from which to select the best combination for recreating the hybrid plant in question. And since transgenesis was used in the first place in this technique, the parentals that will be selected will also have to be rid of transgenes. And ultimately, the null segregant parentals, as well as the resulting hybrid, will have no novel combination of genetic materials. And next is agroinfiltration. As we all know, agrobacterium is widely used in genetic transformation, specifically for transforming calli or undifferentiated plant cells. In this NBT technique, agrobacterium is also used to deliver vector constructs, but this time into differentiated tissues such as leaves, flowers, and immature embryos. In agroinfiltration, the possibility of forming novel combination first depends on the type of target tissues used, namely germline or non-germline, and the type of vector construct used, namely if it is meant for expression only or for plant transformation. For non-germline tissues like ordinary leaves, transient expression or expression of the virus particles is usually the case, and the vectors will not be carried on to the next generation. However, for germline tissues like flowers and immature embryo, the use of transformation vectors will result in the stable integration of E gene into the plant genome, which will be carried on to the next generation through seeds. Now, if the gene introduced is from a non-cross-compatible species or a cross-incompatible species, the product will be a GMO. However, if the gene introduced is from the same species, there will be no novel combination of genetic materials in the final product, so non-GMO. And the last one is synthetic genomics. This is the engineering and manipulation of an organism's genetic material on the scale of the whole genome, based on technologies to design and chemically synthesize pieces of DNA and to assemble them to long chromosome-sized fragments, and such technology includes Gibson assembly. As an NBT, synthetic genomics can create non-GMOs by faithfully duplicating a naturally existing genome, like the genome of COVID-19 virus, for instance, to create the actual COVID-19 virus itself from the genomic sequence data or digital sequence information. It can also create a GMO by introducing slight modifications or trans-like inserts to the genome being assembled. And among the plant breeding innovations, it is gene editing that is gaining ground in the Philippines and in most other countries because of its simplicity and its precision also. In fact, in the Philippines, IRI was already able to publish different papers starting 2017 about their genetically edited or gene-edited rice with regards to efficient water use, and then for the resistance to diseases like tungro and also bacterial leaf blight. And also in the Philippine Rice Research Institute, we're also now producing some putatively edited lines of rice with resistance to tungro and BLB, and other institutions like the IPB or Institute of Plant Breeding, they also have gene-edited eggplants. So this will have a very great impact later on in Philippine agriculture. And now I would like to give you some insight into how the different products of plant breeding innovations are regulated globally. Let's begin with the Philippines. The Philippine perspective or the Philippine perspective is that all of the mentioned plant breeding innovations are capable of producing a non-GMO as a final product. As you can see here in this figure, this can be seen under PBI case 1 on the third column, which are regulated as non-GMOs. And there are some plant breeding innovations that can deliberately produce GMOs. And these are under PBI Case 2 in this illustration, which are regulated under the Joint Department Circular. Now let's go to Argentina. I just superimposed the case of Argentina on the Philippine framework for simplicity and for easy understanding. This is not 100% accurate, so just focus on the highlighted or the red text, okay? For Argentina, SDN is case by case, unlike in the Philippines, which is automatically under case 1. So that means SDN2 can also be considered as a GMO in Argentina. And the products also of synthetic genomics are, and also cisgenesis are considered GMOs in Argentina. And then for Canada, it doesn't matter what technique was used in producing the crop. They're just focused on whether the plant has novel traits. They don't care with how the plant was developed. They only look at the novelty of the trait, meaning if the trait is not present in natural populations of their species, it must be a GMO. Now, for the US, they don't care if genetic transformation was used, for as long as the plant or plant pests are not involved in the creation of the product. So a GMO can be regulated as a non-GMO in the case of the USA. So they are in stark contrast with Canada. So sometimes there are some trade problems with regards to gene-edited or products of modern biotechnology. Now for Australia, SDN2, SDN3, and ODM and cisgenesis are all considered as GMOs, while grafting with GM material is case by case. And now, in the case of Japan, SDN2 is case by case, and ODM is to be considered as a GMO. And let's move on to New Zealand. In New Zealand, actually, SDN1, which is in most countries regulated as non-GMO, is considered as GMO in New Zealand. So when SDN1 is regulated as a GMO, this shows that the regulation is very strict, and other techniques would also most likely be considered as GMOs. And also in the European Union, SDN1 is considered as a GMO, SDN3, ODM, cisgenesis, intragenesis. So they're also very strict in the EU. And recently, there have been talks on how to relax this, and we just hope that everything will just turn out in the favor of the developers. And here are some key takeaways from my presentation. There are different new technologies available to the modern day breeders, and one of them is plant breeding innovations. Genome editing is the most popular among plant breeding innovations. Enabling policies are important for new technologies to have an impact on agriculture. And there are different perspectives globally on how PBIs are assessed. And some countries are lenient, some countries are strict. So that's all, and thank you so much for your attention.
Okay, thank you very much, Dr. Ordonio, for discussing the new breeding technologies that we have now that we can use in improving crop traits, and also for providing an overview of the regulatory framework of countries and how they treat PBIs in their regulation. Again, I will encourage everybody the participants to put your questions in the Q&A box, and we will address them during the open forum. Now we will proceed to the next topic, and this will be on regulatory status of plant breeding innovations in the Philippines. This will be presented by Ms. Jeronima Eusebio. She holds the position of supervising agriculturist and served as the second presentation, sorry, will be by Dr. Gabriel Romero on biotech approaches to mitigating pesticide use in agriculture. We will have Ms. Jeronima later. And so Dr. Romero is the Executive Director of the Philippine Seed Industry Association. Prior to that, he was the Senior Regulatory and Scientific Affairs Lead of Monsanto Philippines for 10 years. He also served as Gene Bank Manager, Director of Crop Biotechnology Center, and Acting Deputy Executive Director for Research at the Philippine Rice Research Institute. He holds a PhD in genetics from University of California, Davis in USA, and a Master of Philosophy in Plant Breeding from the University of Cambridge in UK, and a BS Biology from the University of the Philippines, Los Baños. Among his awards are the Outstanding Young Man, Outstanding Young Scientist from the National Academy of Science and Technology, and Faces of Biotechnology from the Department of Agriculture. Dr. Romero, please, you can now share your slides.
Thanks to the organizers for inviting me here to give a talk on the biotech approaches to mitigating pesticide use in agriculture. I come from the Philippine Seed Industry Association. We are a 33-member strong seed company association in the Philippines. We have multinational seed companies and local seed companies, as well as government and academic institutions as members. So happy to be here to share with you this talk on the mitigating pesticide use in agriculture. So the agriculture enterprise in general is faced with the daunting task of feeding a growing population. We are expected to have a doubling of a very high world population in 2050 that will require doubling of food production. Since the arable land and is decreasing or remains the same, well, actually decreasing because of the industrialization and other developments and conversion. And the number of farmers is also declining. The doubling of the food production will really require efficiencies in farming technologies. So we need to rely on science, research and development to be able to do this. We need to be conscious about the Sustainable Development Goals set by the United Nations. And agriculture is very much a key activity that will help the United Nations or the world achieve zero hunger, the Sustainable Development Goal number 2. We are not yet on track. These goals have been set to be achieved by 2030, but we need to make every little progress possible in order to be able to cope with the big challenge in front of us. And now that climate change is upon us, we are facing more complicated farming problems, especially the advent or the rise of different dynamics of pests and diseases. That's why crop protection is very much part of the strategy to increase food production in the world. That's why we cannot do away with pesticides. Usually people are turned off by the use of pesticides, but pesticides are an integral part of our world, not just in agriculture. But for our topic today, we need to focus on agriculture per se, because this is where the challenge of feeding the world population relies. There are different types of pesticides used in agriculture: herbicides, insecticides, and fungicides. Although for my presentation this morning, I will just focus on herbicides and insecticides. There are various pesticide classifications, and there are different levels of toxicity to the target pests and also to the farm workers or to human health. In general, it's the bioinsecticides exemplified by Bacillus thuringiensis that have the safest that have the best safety profile. And among the herbicides, the phosphonomethyl amino acids, where glyphosate belongs, are also generally classified as very safe. So the question now is, why don't we just focus on the use of these safe pesticides? Well, that is really the intention, but there are special other insects, or there are other weeds that some of these pesticides cannot control. That's why the other probably more toxic pesticides are still being used. But with genetic engineering, we've been able to increase the use of the safe insecticide and also the safe herbicide. This one example is the highly successful BT crops. It's BT, or Bacillus thuringiensis, has been in use in agriculture for more than 50 years. And then genetic engineers cloned the cryprotein genes in Bacillus thuringiensis and put them in the crops. So these BT genes or cry genes are now expressed in the plant itself. There is no need anymore to spray BT spores or sprays on the plant, because that is also less efficient in farming because they are easily washed away. So with genetic engineering, the built-in resistance to the insect has been developed, and this is widely successful. It's not only in corn, but in other crops as well. Soybean, canola, cotton, and others. So actually, with the development of BT crops, in this particular example, BT corn, the use of insecticide has really gone down since the introduction of the BT corn. This is just an example in U.S. cornfields. When the BT corn was introduced in 1996, it has steadily been adopted in larger areas, and the use of insecticide has gone down almost inversely as the adoption of the BT corn. Why? Because the BT corn itself can produce its own insecticide. So there's no need to anymore spray other insecticides that will combat the target pest or the lepidopteran insects in the case of BT corn. And we have that example right here in the Philippines. We've been using BT corn since 2002. So it's more than 20 years now that our farmers have benefited from this technology. Just like insecticides are indispensable, herbicides also are important in farming because they help eliminate the weeds that compete with the crop. So without herbicides, or farmers stand to lose 30 to 40% of their crops Okay, so it is also important to use herbicides, and one of the safest, so far the safest herbicide that is available is glyphosate. It is the most thoroughly evaluated herbicide in the world, and the safety profile is outstanding. Okay. So it is approved by regulatory agencies in over 160 countries. Okay. There are unfounded allegations against glyphosate, but overwhelmingly, the majority of the regulatory bodies overwhelmingly vouch for the safety of this glyphosate. So if we are concerned about herbicide use in farming or in agriculture, why don't farmers or agriculturists focus on the use of the safe herbicides? And that is also how genetic engineering allowed for farmers to focus on the use of a very safe herbicide. So this is the technology behind the use of glyphosate tolerant or glyphosate ready. Well, the brand is Roundup. How this product came about. So glyphosate naturally blocks the EPSP synthase in the plant. So if that is blocked, the amino acids that are important in protein synthesis are no longer produced. So there will be the plant growth will be disrupted. So the plant will die. But with genetic engineering, scientists have been able to isolate a gene from the bacteria, Agrobacterium, that is resistant to the action of glyphosate. So this particular gene is not affected by glyphosate. So what the scientists did was to insert this gene to the plant so that the plant will no longer be affected by glyphosate. And this was made for corn, soybean, cotton, and canola. So these crops are no longer affected by glyphosate, even if the plant or the seedling is sprayed with the glyphosate or Roundup, while the weeds around these crops are killed by the herbicide. So this is a better illustration of the action of glyphosate on the regular plant and on the GM crop, whether corn, cotton, canola, and soybean. The regular crop is also killed by glyphosate along with the weeds around it. But the glyphosate-tolerant crop continues to grow or survive. So this really makes farming very convenient to the farmers, because first, they won't need to till the land as much as before to kill the weeds, to prepare the field for the plants, for the crop, because with no tillage or minimal tillage, the farmers can just sow the seeds, the GM seeds, and let the weeds and the crop grow together. Then after a few days, just spray the glyphosate and all the weeds around the crops will die. Okay. And because of that, the technology has been widely adopted. And with that adoption, there is a lot of environmental benefit, because of less tillage, there is less use of fossil fuel. And because of the adoption also of BT crops, there will be more beneficial insects that abound in the fields, because there are no longer insecticides that also kill friendly insects. So these GM crops are very successful globally. And because of the convenience and the benefit to the farmers, the benefit to the environment, and the increased production, actually, the increased productivity afforded by these GM crops, a lot of countries are now growing GM crops since 1996. This is the data collated by ISAAA. So since 1996, there are more than 30 countries now that are planting GM crops. So, but these are farmer-friendly technologies. And if you come to think about it, they are technologies that mitigate pesticide use. And we are in the map. The Philippines is, I think, number 10 or 12 in the hectareage of GM crops, in the hectareage of GM crops. And right now we are still, we are planting majority of GM crops to GM corn, but soon we will be planting GM eggplant and also golden rice or GM rice, because they have been also recently been approved by our regulatory bodies. So there are already 440 GM events approved since the introduction of the first GM crop in the world. A great majority of them are HT or high herbicide tolerant or IR insect resistant crops. And most of these crops are stacked, meaning they contain both the herbicide tolerance and insect resistance in the same variety. But now we are also seeing the approval of many other events, including the disease resistance traits in crops. So that will also lead to better mitigation of pesticide use in agriculture. So right now we have this global impact of the judicious application of pesticides. That is really the beauty of the application of GM crops. We have contributed to better use of pesticides that have the best safety profiles, and that has led to good reduced environmental impact and also, of course, less impact to human health. In the Philippines, the use of the insect resistant and herbicide tolerant crops, in particular corn, has not only led to better mitigation of pesticide use, but in fact has contributed to the economics of farming. On the average, our corn growers that are using GM crops, GM corn, are earning additional 8,000 pesos per hectare. Now that we are in the era or starting era of gene editing, there are also opportunities for the use of gene editing to develop insect resistance in crops. And there are two ways to do it. We can edit the plant itself or the crop itself, or we can also edit the target insect pests, which may be more difficult. But for the editing of plants, it is possible to knock out the susceptible genes in plants, so that will make the plant resistant to the target pests. And the plant may also be edited to have modified plant volatile blends, so it will not be more attractive to the insect pests anymore. Or the editing may also lead to a change in the color of the leaves that will also become less attractive to the insect pests. And gene editing is also possible to develop herbicide tolerance in crops. And this one is an example of one that is developed in rice. So if you remember, the glyphosate tolerance that was developed by genetic engineering needed to isolate the gene from bacteria. So with gene editing, that is no longer necessary. The rice itself may just be edited through the CRISPR-Cas technology to do some edits on its DNA. And this is an example of a gene editing done in rice that converted the tryptophan to leucine at the position 548 of the acetolactase synthase gene. This protein happens to be disrupted by the herbicide Spiribac sodium herbicide. This herbicide is also has an outstanding safety profile. Maybe not as outstanding as glyphosate, but half of the safety profile of glyphosate. So this is also a safe herbicide. So this promises to be another way of our new technologies, biotechnologies, especially gene editing, to be able to mitigate the pesticide use by focusing on the safe pesticides. So in summary, pesticides are indispensable in farming. We cannot do away with pesticides, but as I showed you, some pesticides can be synthetic or organic, and there is continuous development of more efficacious pesticides with better safety profile. That is really necessary, okay, for the safety of the farmers and the consumers. And genetic engineering already created highly successful and widely adopted GM crops containing the safest insecticide, and that is Bacillus thuringiensis, originally only used in organic farming, and compatible with the safest herbicide, just like glyphosate. And gene editing is now at the threshold of a new wave of products that can further mitigate pesticide use in agriculture. So with that, I'd like to thank you for your attention and be happy to participate in the Q&A later. Thank you very much.
Okay. Thank you very much, Dr. Romero, for highlighting the importance of crop protection on agricultural productivity and also on the use of biotech crops to promote judicious application of pesticides that contributes to beneficial environmental impact. So we will now move the last presentation on regulatory status of plant breeding innovations in the Philippines. This will be presented by Ms. Jeronima Eusebio. She holds the position of supervising agriculturist and serves as the OIC head of the Bureau of Plant Industry Biotechnology Office, the central hub for GM applications and biosafety permit issuances. Additionally, she chairs the Biotechnology Core Team Plant Breeding Innovations Group, responsible for conducting technical consultations to assess and determine the regulatory status of PBI products. Ms. Eusebio is also a deputized plant quarantine officer with prior experience as the head of National Plant Quarantine Service Division, Central Laboratory for Pests and Disease Diagnosis. She has represented the Philippines as delegate to the ASEAN Task Force on Genetically Modified Food Testing Network, or GMFNet, and served as the national focal point of the... can you please kindly mute your... Okay, let me continue. Her academic credentials include a Master of Science degree in Agricultural Plant Biotechnology from the Hungarian University of Agriculture and Life Sciences in Budapest, Hungary, as well as a Bachelor of Science degree in Plant Pathology from the University of the Philippines, Los Baños. Emma, you can now share your slides.
Good morning, everyone. I would like to express my gratitude to the organizing committee for extending the invitation to discuss the regulatory status of plant breeding innovations in the Philippines, or the Philippine policy for gene-edited products. Okay. Okay. Next. Let me provide an overview of the policies development. In 2018, the Philippine Department of Agriculture initiated the study titled A Review of the New Plant Breeding Techniques from a Regulatory Perspective. This study was conducted by a team of respected scientists and regulators. The primary objective of the study was to assess whether the application of NBT would result in products with novel genetic combinations. The findings of this study laid the groundwork for policy recommendations. Then the study was subsequently presented to the National Committee on Biosafety of the Philippines, as they are responsible for developing biosafety policies within the country. Following this, the NCBP issued Resolution Number 1, Series of 2020, titled The Regulation of Plant and Plant Products Derived from the Use of Plant Breeding Innovations or New Breeding Techniques. Okay. These are the key resolutions from the committee. PBI products can be categorized as either GMO, which contain a novel combination of genetic material through modern biotechnology, or non-GMO or conventional product if they lack a novel combination of genetic material. If PBI-derived products are deemed GMOs, they will fall under the regulation of Joint Department Circular 2021. However, if PBI-derived products are not GMOs, they will not be subject to JDC regulation. And the third one, the Department of Agriculture is designated as the lead agency responsible for evaluating and monitoring PBIs or NBTs. The resolution was signed by 7 department secretaries and CBP scientist members and organization representatives. In accordance, so this is the Memorandum Circular number 8. This covers rules and procedure to evaluate and determine when products of plant breeding innovations are covered under the DOST, DA, DNR, DOH, DILG Joint Department Circular number 1, Series of 2021, based on the NCBP Resolution number 1, Series of 2020. Okay. So in accordance with NCBP Resolution number 1, Series of 2020, PBIs or plant breeding innovations are defined as novel array of molecular tools that facilitate the precise and efficient development of new crop varieties with desired characteristics. These tools are notably more precise and expedite the breeding process. According to the policy, the PPI products can be categorized as either GMOs or non-GMOs, which are conventional products. Product is classified as GMO if it contains novel combination of genetic material. Conversely, if the end product lacks novel combination of genetic materials, it falls under the category of non-GMO or conventional. PBI product with novel combination of genetic material will be regulated under JDC 1, 2021. Okay. So the technical consultation for evaluation and determination, or what we call TCED, is a technical evaluation of the PBI product to determine whether the final product of the PBI used contain a novel combination of genetic material, and therefore whether the PBI product falls under the coverage of JTC 1 2021. The conduct of the technical evaluation and determination on the regulatory status of PBI product shall be the responsibility of the TCED group, which shall be composed of at least 2 members of the Biotech Core Team Plant Breeding Innovation Group and an external expert if necessary. So a product developer, which you can see the list here, may include the department, university, and so on, may submit a TCED request. For instance, in the case of non-resident product developer, the TCED request can be submitted through a representative residing in the Philippines. This illustration provides an overview of the process of determining the regulatory status of a PBI product pursuant to DA-MC 2022. The entire process takes approximately 27 to 32 working days. It consists of 3 phases with detailed explanation to follow in subsequent slides. First one is the pre-assessment phase, which is completed within 3 working days. This is being done by the Biotechnology Secretariat. And the second phase involves the technical consultation, evaluation, and determination spanning 24 days, which coincides with 10 working day public consultation period. And the final phase necessitates 5 working days for the official determination of whether the PBI product is classified as GM or non-GM. This slides provides an overview of the procedural prerequisites for TSED. The first step is the submission of a request for TSED with supporting documents. You can see the submission requirements listed in this slide. The printed electronic copy of the TSED request form, accomplished prior evaluation form, and the scientific studies, experimental evidence, and other documents to support the claim in the PEF. The TSED request form reflects essential details about the product developer, while the PEF contains information on the type of organism and species involved, the PPI technique used, novel characteristic introduced, and evidence of the desired genetic changes. The BPI Biotechnology Office shall then review this submission and it is considered officially accepted when it meets the required criteria in both form and substance. Once accepted, it will be forwarded to the Biotechnology Core Team Plant Breeding Innovation within 3 working days to constitute the TSED that will evaluate the request. Okay. Here is one of the requirements for the submission, the TSED request form. This form encompasses essential information such as the name and contact details of the product developer, and when applicable, an appointed agent. It also mandates a sworn statement from the agent affirming the authenticity and accuracy of all submitted documents. Okay. Here is the second form required, the prior evaluation form, which provides a more comprehensive breakdown of the plant breeding innovation procedures employed, like what Dr. Antonio mentioned earlier, the SDN1, SDN2, SDN3, and others. Then they have to identify the sequences of the guide RNA, the delivery system, whether it's agro or other methods, and then nucleotide sequence to be introduced if applicable, and other relevant information. As you can see, this form contains a section to be answered by the TSED group. This is where the technical determination of the TSED group shall be indicated. So we have to affix our signature in this form after final determination of the regulatory status of the PBI product. Okay. This slide outlines this process of conducting TSED. So following the official acceptance of the submission, the BCT-BPI shall constitute the TSED. This group includes a minimum of 2 experts from the BCT-PBI, if required, an external specialist. In the determination of the regulatory status of, in case of the reduced browning banana, we engage an external expert to participate in the evaluation. And the primary objective of the evaluation process is to determine or to discern whether the PBI product consists of novel combination of genetic material. If that is said, requires clarification during the discussion or during the evaluation, the product developer might be asked to participate in the deliberation process. At the conclusion of the said, that the said group shall make its technical determination, and the Biotechnology Office shall then endorse the technical determination within 7 working days to the BPI director. The entire evaluation process is designed to be conducted within 24 working days. Okay. So Dr. Aldon, you already presented this decision tree. Actually, this is an annex of the NCBP Resolution Number 1, which shall be used by TISED as guide in making technical determination on the regulatory status of the PPI. So I will no longer discuss this one since Dr. Aldon, you already extensively discussed this figure and this flowchart. Okay. So this is again the portion of the PEF or the prior evaluation form where the TISED group shall indicate its technical determination. After the conduct of the TISED group shall determine whether the PPI product does contain a novel combination of genetic material, at which case they will check the second box, or does not contain a novel combination of genetic material, which case they will check the first box. The public commenting period is another step during the processing of submission. Once an officially accepted request is received, is posted on the Bureau of Plant Industry website within 10 working days to gather feedback from the public and to ensure transparency in the process. It is important to note that only technical comments will be taken into account during the final determination of the request. The last step of the process is the official determination of the submission request by the BPI director. The director has 5 working day window to make a decision after receiving recommendation from the said group and technical input from the public. The regulatory status of the BPI product is officially determined as GM. Then the product developer will be advised to proceed with their application under JDC 1, 2021. If non-GM, they will be given a certificate of non-coverage. And the CNC is made public by posting on the BPI website. So this is the certificate of non-coverage if the determination is non-GM. Here are some important notes on the certificate of non-coverage. It shall refer to the novel characteristics introduced in the current variety and the subsequent progenies. It applies to all germplasms of genetic backgrounds that will contain such characteristics produced by the product developer or its licensing in further breeding. And most importantly, the certificate of non-coverage does not excuse the product developer from complying with other relevant regulations of the Department of Agriculture and other government agencies such as those involving plant quarantine, pest risk analysis, varietal registration, and crop-specific standards and programs. The policy also includes a provision for appeals. If a party feels aggrieved, they have the option to file an appeal with the Department of Agriculture Secretary. This appeal must be submitted within 15 working days following the posting of the determination decision on the BPI website. So what are the salient points of the policy? It prescribes a product-based approach on the determination. There is a recognition that PBIs can generate both GM and non-GM plants as products. Hence, the evaluation and determination focuses on the nature of genetic change that is present in the product. This is in contrast with the process-based approach where products of a certain PBI are immediately determined as either GM or non-GM by virtue of the employed process technique alone. And second, it is not a regulation on product safety, whereas the JTC 2021 employs the job for the assessment of the safety of GM plant for human, animal, and the environment, the DAMC 8 task the research group to determine the PPI product, if it is GM or not GM. And then there are two types of determination of the regulatory status of PPI. The technical determination is made by DSED, while the official determination is made by the collector of PPI. In making the official determination on the regulatory status of PPI product, the director considers both the technical determination, which indicated in the complete spec by the DSED, and the technical information from the public during the commenting period. Okay. So if you have further question, you can reach us both through this contact information. I think this is my last slide. Thank you very much for listening.
Okay, thank you very much, Emma, for providing an overview of the guidelines in the assessment of products of PBIs, and for emphasizing that the current regulation on PBIs is not a regulation on product safety, but rather the evaluation and determination if the PBI is a GM plant or a GM product. And
Yes.
then GDC will take care of the evaluation of the safety of GM plants, if it is a GM plant. For those who are asking about the presentations, we will make available the presentations and the recordings in the ISAAA website, of course, after the completion of the webinar series. So you have to wait until we completed the series and until day 3. So we will now proceed with the open forum, and we have quite a number of questions in the chat box. And there is one question here from an anonymous attendee, and we acknowledge our participants from outside the country. There's a question here about GMO products, if they can harm our daily intake. Can anybody from the panelists answer this? Maybe we can ask Dr. Ordonio, and Emma can supplement the answer. Dr. Ordonio, please. This is a very basic. I think this is a very basic question. Is GMO food products can harm our daily intake? I don't know what that means.
Part and parcel po ng GM regulation, particularly in the Philippines, is to ensure the safety of our GMO products in terms of human health, animal health, and the environment. And we have a rigorous biosafety process. It has been established since year 2020, 2000, 2002, and earlier. And we have 30 decades actually of biosafety regulation in the Philippines. And because of that, our regulatory system is very much functional, and we always focus on the safety. The fact na na-approve siya, the fact that it was approved by different regulatory agencies during the process, then it would certify that it is safe indeed.
Okay.
Yes.
Can you add to that, Emma?
Yes. We have international protocol or standards being followed in assessing the safety of these GMOs. So we are signatory to international bodies to ensure that GMOs that we introduce to our consumers, to the public, are safe. So based on the applications that we've received, we have issued biosafety permit indicating that they are safe. So it's case-to-case basis depending on the submission of the technical documents submitted to the regulators. And it will be the basis in assessing the safety of these GMOs for human, animal health, and the environment. Thank you.
Okay, thank you, Emma. So another question from our participant. How can we strike a balance between promoting the adoption of innovative plant breeding methods and ensuring they do not harm biodiversity or have unintended ecological consequences? Anybody from the panel can answer this one? Maybe Dr. Gabby, since you discuss about some environmental benefits on the use of the biotech crops. So the question is, how can we strike a balance between promoting the adoption of innovative plant breeding methods and ensuring they do not harm biodiversity or have unintended ecological consequences? Dr. Gabby?
Okay. The question may also apply to GM crops.
Mm-hmm.
So in terms of harming biodiversity, that is not really what happened with GM crops. Because with the use of BT crops, let's say BT corn, the cornfields have become more have become more friendly to other insects. Yeah, there are now more non-target insects that abound in farmers' fields. Unlike before, when insecticides against corn borer was heavily used, the non-target insects are also affected. So in terms of the biodiversity, in fact, the GM crops have increased biodiversity. And in terms of ecological consequences, GM crops are also the herbicide-tolerant crops, in fact, have contributed to less use of fossil fuels. So it's been in record that it's like removing I think thousands of cars from the roads because of less use of fossil fuel, because there is no longer much cultivation or land preparation using fossil fuels, diesel, etc. So the question is not really accurate, because GM crops, and now we are looking at gene editing as even a more efficient technology, they in fact contribute to increasing biodiversity and reducing or actually avoiding ecological or impact to ecology or to the environment. So I think we really need to rethink about our assumption that GM crops are harmful to biodiversity, because our experience with GM crops is they in fact contribute to biodiversity, and they also help prevent ecological disasters.
Okay. There is a follow-up question, and maybe this is addressed to Dr. Ordonio, since he discussed about the new wave of available new plant breeding tools. As climate change continues to impact agriculture, how do you foresee these new plant breeding techniques contributing to the development of climate-resilient crops and sustainable farming practices? Dr. Ray?
Yes, in this era of climate change, we have really breed crops in the soonest time possible. We have to release them in a relatively shorter period of time, and that's where gene editing comes in handy, because we can develop crops in less amount of time compared to conventional breeding, and it's even more precise. Like in the case of drought resistance, probably later on we can identify some genes and we can tweak or gene edit that in the particular crop that we like, and then we can probably reduce the time in the development of such crops. Drought tolerance, for example, for the grain yield in the face of climate change. It's very useful, I think.
Okay, thank you. There is another question maybe you can also answer. This is a live from our FB Live posting from Miss Karen Barondok Albiar. And the question is, how do we reconcile using non-GMO products produced from processes using recombinant technology for use in organic agriculture. The new PBIs for feed or food ingredients is inevitable. I think using products classified as non-GMO should be considered for organic agriculture as well. Ray?
Yeah. Okay. If I got the question correctly, in the Philippines, we have RA 100 or 10068, or the Organic Agriculture Act of 2010, in which it defines genetically modified organisms as not fit in the standard, right? And so golden rice, for example, is considered as non-organic. It is not possible in organic agriculture. And that act was enacted in 2010. And after that, Davao created a resolution in their city prohibiting coexistence of GM and conventional crops. And other municipalities followed suit. Even the Science City of Muñoz now has that organic agriculture resolution. And actually, in the original Organic Agriculture Act, there's no mention of the non-coexistence. Only in the Davao resolution was it there. So it was then copied. So we just hope that for the other municipalities, there won't be that coexistence principle in there. They can actually make it more lenient, because the DA is both promoting organic agriculture and the standard conventional agriculture. Both of those are beneficial to our country if we can just use them properly. So because of that, I don't have any problem with organic agriculture. It's just that existence of both must be allowed. And with the gene editing, the beauty of gene editing is that later on it will be approved as a non-GM, and the more that it will be acceptable to Filipinos. Thank you.
Can you add anything, Emma, about organic farming and the use of GM crops? Emma?
Yes, the DA has both, Dr. Ordonio already mentioned that DA has both policy on organic farming and the use of promoting the use of products of modern biotechnology. So there are areas in the Philippines that promote organic, and they don't want the entry of this GMO. So the the regulators also respect this preference of the local government units if they don't want GM products in their locality. Actually, it is one of the conditions stipulated in the Biocity permit that the technology developer has to respect that ordinances existing in certain locality. Yes, thank you.
Thank you, Emma. There's a question, I think this is addressed to Dr. Ordonio, from William Tebes Jr. How does gene editing of rice impact its nutritional content? And additionally, if this technology is commercialized, how will it affect pricing? Ray?
In editing, it depends on target gene. If the gene is involved in nutrition, it will have an impact on the nutrition value of that crop. But otherwise, only the traits that are not about nutrition will be affected. So it really depends on the target gene. That's why it's called precision breeding, because we know the target gene and we know the eventual phenotype of such plant or variety. And when it comes to commercialization, it should be the same with the ordinary variety in terms of price, in terms of how you plant it. Everything should be the same except for targeted trait.
Okay. And I think this is a question about insect resistance. Majority of our farmers now using GMO product are using GMO product. How comes that they are experiencing infestation of fall armyworm around Isabela area? Who can answer this one? Doc Gabby, do you have any experience about army polworm infestation in the area of Isabela?
Yes. Thank you for that question. Fall armyworm is a new pest that came to the Philippines, I think, 3 or 4 years ago. So, and it is ravaging our crops, especially, I think onion especially, and to some extent, maybe a considerable extent, corn. So the insect resistance in BT corn, in the present traits in our corn varieties and our corn hybrids, some of them can control fall armyworm, especially the ones that have two cry genes. The varieties or the hybrids that only contain one cry gene or BT gene is somewhat susceptible. So, and yeah, maybe you saw that in the farmers' fields. That's why there is really a need to have continuous development of insect-resistant crops, insect-resistant corn. This is really a serious battle against fall armyworm. But fortunately, I think there are now available corn hybrids that I said contain at least two BT genes that can control, that can manage this fall armyworm. And hopefully our farmers shift to the use of this stock or these pyramided corn hybrids, so that the fall armyworm will be stopped in their fields. And hopefully the research-based companies continue to develop really efficacious insect-resistant hybrids, because we don't know whether fall armyworm will be the last to be introduced as a new pest in the country. There may be some new insect pests that may come. So, and because of climate change, this is a distinct possibility. That's why we need to empower our R&D institutions, also facilitate also empower research-based companies, because this is a continuing battle against pests and diseases in our farmers' fields. But that's true. That's true. Our insect-resistant hybrids, especially the ones that have only one cry gene, seem to yield, or succumb to fall armyworm.
Okay. And I think this is the question is connected to the previous question. Does spraying BT, the organic pesticide, to BT corn makes the fall armyworm more tolerant?
I don't know, but we can try. There may be multiple actions or mode of actions of the BT sprays. So we have to explore. What I know is the GM ones with multiple BT genes are resistant to fall armyworm. So yeah, the farmers can explore both the BT sprays and even the use of the GM corn against fall armyworm. And there may be companies that are companies that may be offering BT sprays that are really good against fall armyworm.
Okay. And I think this question is addressed to Dr. Ray. Is it possible that in the near future, crops would be able to mutate their own genes without the use of breeding techniques to increase its tolerance to pests and other diseases? So instead of doing gene editing.
You know, Sir Pompe, there is such thing as a spontaneous mutation. And this goes hand in hand with evolution. And you know, evolution takes place in millions of years. It's happening up to now, actually. And that's the reason why we are, improving plant breeding innovation tools, is to speed up the process of modifying the genetic makeup of plants such that they can be more useful to us. If we just allow them to mutate by themselves, then it will be too late when the time comes that we have to address climate change, for example. That's why we have to really invest on gene editing technologies and other new technologies.
Okay, thank you, Ray. This question is, I think, anybody can answer or everybody can answer. Based on your assessment, how deep is the understanding of the grassroots, including the local government units, about GM crops? Emma, can you provide the first answer?
Yes. Actually, in areas wherein there is high adoption of GMO, they they know very well GMOs. But though in those areas that like BT corn, because we introduced first BT corn, there's very limited knowledge about GMOs. So in those areas, we have to strengthen the our information campaign about the introduction of this technology and how they will benefit from using this technology. So pretty well they know in those areas with high adoption of GM crops. But there are also areas where in very limited knowledge on the use of this technology. Okay.
Ray, how about understanding of the grassroots? About the question is about understanding of the grassroots.
I completely agree with Ma'am Emma.
Okay.
With regards to that, yeah, because of our Golden Rice project, we were able to go to different places, and yeah, for majority of areas, they are not yet aware of what GMOs are, and sometimes because of the lack of awareness, they issue resolutions against GMOs when in fact, when asked, they are actually in favor of GMOs. So there's that disconnect between what they think and what they do in terms of writing resolutions, for example. So we really have to intensify our outreach to them, to educate them, awareness campaigns like that.
Okay, thank you. I don't know who can answer this one. How does the distinction between GMO or non-GMO relate to intellectual property rights, specifically patents? I'm not quite sure who can, or maybe Ray, can you answer this one? How does the distinction between GMO or non-GMO relate to intellectual property rights, specifically patents?
Yes, for GMOs, yeah, private companies are being involved there, and it's something to be guarded. I mean, the IP is something to be guarded in their case, and that's actually very, I mean, very important in the case of private companies, the patents. For gene editing, the same is true. Actually, it's not yet clear for us regarding the commercialization of gene edited crops later on. As of now, we're using vectors. They're licensed to us for research purposes only. But then later on, by the time that we are already going to commercialize our crops, still another hurdle is how to get the license for commercialization. So that's an issue in terms of gene editing.
Okay, thank you, Ray. And I think the question is for Dr. Gabby. Is there, aside from the yield guard and the stock trait Roundup Ready corn, are there any other corn varieties that will be available in the market?
Yeah, definitely. These companies are working in earnest to develop new insect-resistant varieties. I just don't know what events they are creating, but it's also their strategy to combine the existing tri genes or also BIP genes So all these approved insect-resistant genes may just be combined. And this combination is already a powerful new product that can be sold in the market. So just expect these companies to be introducing new products, but they still have to hurdle or pass our regulatory agencies. That's a hurdle that they need to pass, and that also require some time, maybe 2 or 3 years in the process. But the starting materials are already there to combine these approved existing genes, and aside from these existing genes, they are also working hard to use gene editing to also produce new insect-resistant genes that can be put in corn and other crops. So yeah, expect that there will be new waves. Well, hopefully the regulatory bodies may also be able to cope with these applications. So yeah, maybe I call to the government to provide more funding to BPI, more people, so that these applications can be processed fast, so that our farmers can already will benefit from these new products.
Okay, thank you very much. There's a question here about use of irradiation in new plant breeding innovations. What is the measurement for the level of irradiation to assure safety in plants?
No, radiation is not involved in any of the new plant breeding techniques. I think you're referring to mutagenesis, and that's not plant breeding innovation. That's a conventional plant breeding that is being used since the 1920s, like that.
Okay, and there's a question about the contribution of biotechnology innovations on environmental impact reduction in agriculture, and I think this has already been answered by Gabby. Another question, in relation to GMO crops and human health, why are some countries ban GMO food products and some of the consumers look for non-GMO certified products? Anybody can answer that question?
Okay, it's a matter of preference, Sir Popen. And it's not actually totally banning the GM. In Europe, they ban the planting, but they still import GM products. So they still use GMOs for processing. They still source from other countries, from US, raw materials to process their food. So it's not a total ban, but more on planting in EU, and in other countries, majority it's for planting ban in EU, but they still use GMs as raw materials.
The question here now is, how can smallhold farmers better access these new plant breeding innovations? Any suggestions from our panelists?
Right now, we just had the first policy on gene editing approved here in the Philippines. So I think this is the start. In fact, there was already application approved by the DA-BPO, the tropic non-browning banana, right? And I believe more and more crops would follow suit. There will be additional applications later on, like rice, for example. And I think the environment now is more conducive than ever towards the proliferation or the commercialization of modern biotech crops.
Okay. Another question is there a possibility to emerge new bacteria or insect due to mutation, adapting to the capability of GMO crops? Anybody can answer, or Dr. Ray, can you answer? Is there
Yeah,
a possibility?
it doesn't matter whether it's a bacteria or other organism. You can just get the DNA. What matters is the gene. You can take it from any organism, and then doing genetic engineering, you can transfer it to another organism like plants. As long as you comply with the regulatory process, then you're fine. It's okay.
Mm-hmm. Okay. I think we have covered quite a lot of questions. And thank you very much, panelists, for your answers, for giving clarifications to the questions. It's already 10 before 11, and I think I need to provide a synthesis for today's activity. For the certificates of appreciation to our panelists, we will send it to you via email. Thank you very much. And I think, can I ask all the participants to give a round of applause, virtual applause, to our panelists, to our resource speakers. Now, I proceed with the synthesis. This webinar series is envisaged to contribute in promoting and enabling environment for the adoption and use of biotech innovations in transforming Philippine agriculture as a productive and sustainable sector. As underscored by Dr. Mingala in his key message, there's a need to address daunting challenges in agriculture. Biotechnology is recognized as a game changer with the potential to address these challenges and revolutionize food production system. The first presentation by Dr. Ordonio discussed the different plant breeding innovations and their potential impacts on agriculture. Advances in knowledge about specific gene functions and plant genome, data analytical tools such as bioinformatics, high-throughput genotyping and phenotyping tools, and DNA synthesis technologies, among others, provided the needed tools for new plant breeding innovations. New plant breeding tools include site-directed nucleases, oligonucleotide-directed mutagenesis, cisgenesis and intragenesis, RNA-dependent DNA methylation, grafting with GM material, agro-infiltration, and synthetic genomics. Genome editing is the most popular among the PBIs, considering targeted and efficient genetic changes to produce beneficial traits. It is emphasized that enabling policies are important for the adoption and use of plant breeding innovations. The second presentation by Dr. Gabi Romero highlighted the importance of crop protection on agricultural productivity and sustainability. The use of pesticides, such as fungicides, insecticides, and herbicides, has been an integral part of crop protection regime, enabling plants free from diseases, reduction in crop losses, and protection against weeds. The use of biotech crops promotes judicious application of pesticides and contributes to beneficial environmental impact. Gene editing offers new tools for developing crops with insect and disease resistance and herbicide tolerance. The last presentation of Emily Eusebio provided an overview of the guidelines in the assessment of products of plant breeding innovations. The evaluation and determination of PBIs focuses on the nature of genetic changes in the product. The current regulation on PBIs under DA Memorandum Circular Number 8, Series of 2022, is not a regulation of product safety, but rather the evaluation and determination if the PBI is a GM plant or a product. JDC number 1, Series of 2021 covers the evaluation of the safety of GM plants for human and animal health and the environment. So in summary, the key takeaways from the three presentations are the following. There's now an array of new breeding techniques or plant breeding innovations available for improvement of crop traits. Gene editing is becoming the preferred tool because of targeted and efficient genetic changes to produce beneficial traits. The use of biotech crops promotes judicious application of pesticides that contributes to beneficial impacts to the environment and Department of Agriculture MC number 8 series of 2022 offers guidelines on the regulatory pathways for products of NBTs or PBIs, while JDC number 1 series of 2021 covers safety evaluation of the GM plants. With that, I thank you all the participants and our resource speakers for this webinar, and we can conclude this webinar. I hope we can see you again tomorrow and on day 3, and you can complete the webinar series. You have a parting words from Dr. Aldimita, please.
There are still a lot of questions, very minor ones that were not addressed. So email us. A lot of them are dealing still with GM and biosafety. We can raise them up tomorrow, although tomorrow it's going to be focused on animals. Today it's on crops. Also, there are also questions about the availability of PowerPoints, and we have already, and Sir Popen already told us a while ago that it'll be available next week as soon as all of the presentations are over. So the webinar is still Friday, and we will have it ready by next week. The certificates will be available also on Friday as we finish the webinar series. And if you respond to the evaluation survey at the end of the third webinar. Okay. So thank you so much to all our speakers. Thank you so much, Sir Popen, for a wonderful moderatorship. And we hope to see you. You will be using the same link so that you won't get lost. And I hope to see you all. We were able to have around 450 attendees today. And you can also invite all your friends to come over, and so that we can discuss now about biotechnology, something new to the Philippines. Have a great day, everyone. Thank you so much.
Thank you.
Thank you po.
Bye. Bye-bye.
Bye.
See you all again tomorrow, all the participants today.